visible.
Okay, so where were we in the last class? Where are your batchmates? Are they not joining up? Is it still Eve there?
No, sir.
Oh, right. They should have joined by now. Yes, so let's move forward. They are missing out on today's class. Okay. Okay, what did we do in the last class? Just a brief recap since it's you and me only. So, will you please give me a recap?
Yes, sir.
Yes.
We studied like self-pollination and cross-pollination,
Yes.
like types of pollination is studied, and we studied pollinators. What are pollinators? There are two types of pollinators: biotic and abiotic pollinators.
Yeah.
Living pollinators like birds, insects, and honeybees. We took example, and in abiotic we took air, water, and wind. That's
Yes.
and,
Will you go back? Okay. Yeah. Continue.
and in cross pollination, like in biotic, in self pollination, we took autogamy. Like auto means self, and gamy means like fusion, union.
There are three.
Okay, like autogamy basically means like the pollination within this same flower. It can be of two types: cleistogamy and some one more was there. I read cleistogamy and.
Did you go and figured out at least examples for these three types of pollination?
I think for getinogamy, I saw like tulip, something like that. I don't remember it.
Okay, so you have to find examples and write it down, and also let me know, because this was a part of your homework, right? In class.
Yes.
So you have to do it. Okay.
Okay. So getinogamy basically means like the pollination within the same flower, within the same plant, but different flowers of the same plant.
Yes.
So yeah, and the xenogamy basically means if there are two different flowers but of same genes, so they can pollinate each other. Okay.
Yes, perfect. You also used a word which I was going to tell you later on after I'm done with the seed, right? Clisto.
Yeah.
Right. So did you also read about that?
So cleistogamy, I didn't read in like brief, but
what does the
one more was there: cleistogamy and zetan.
This means
it's closed, something like that. Closed.
Means the
closed flower.
Closed fist is sometimes also known as. Okay. So the concept called closed flower, a flower which never opens to the outside world. Okay.
Yes.
And in cleistogamy, will there be pollinators then?
No, sir. No need of pollinators.
Then how will fertilization happen? Only self-fertilization will happen?
Yes, sir. Self-fertilization.
Oh, yes, right. Okay. Then went on to the second stage, which is formation of zygote.
Yes. So, in formation of zygote, basically when from anther, pollen is transferred from anther to stigma. Then in stigma, the pollen germinates and forms. Pollen fertilizes.
Pollen makes a tube. Germination. The word germination is used with respect to seed, not with the pollen. Okay, or spores.
Yes.
Spores and seeds can germinate, but pollen, it actually forms a tube inside, which is called pollen tube. Some books write it as
Uh,
eating pollen tube, but yeah, yes, continue.
okay. So first I will describe like what is pollen and its layers. So outer layer is known as exine, inner layer is known as intine, and one well, we can say like the exine is attached to the inner layer from germ spore. We can say like germ spore like that.
We'll come to the outer layer in today's class. Okay, so you can skip the outer and inner layer. You can directly come to the generative and vegetative cell.
Okay. So after after zygote formation, the pollen grain forms two divisions. After mitotic division, it forms generative cell and vegetative cell. In generative cell, there are two male nucleus. In vegetative cell, they are uh, it helps vegetative helps it helps in formation of like pollen tube. I think so.
Yes, and then
Yeah.
basically,
And then it forms embryo. It forms an embryo,
which nucleus leads in a pollen tube?
which can you repeat your question, please?
Which nucleus, vegetative or generative, which leads in a pollen tube?
Pollen tube vegetative.
Yes, right. Because it forms the pollen tube, therefore it is leading, and both the
Yes.
male nucleus are following it. Okay.
Hmm. So now we studied the structure of embryo, embryo sac.
Yes, the embryo.
Yes.
That also in angiosperms. Okay.
Yes, sir. We studied sorry, disturbance was. Okay. Uh, in structure of embryo sac, we studied it is seven-celled and eight nucleated structure. Three antipodal cells are there in the center. Two polar nuclei are there. One egg cell. Two synergate cells. Then we studied. Yeah, we studied the structure of embryo sac. First, there is micropylar region, micropylar end. Then it comes integuments, the structure which encloses the embryo, basically. Yeah.
Yes.
Then can you start like again funicle, like first funicle, then
Not yet. Not yet.
okay, not yet.
We have not done the fertilization thing yet, right? The double fertilization thing. After that, we'll study seeds, and then we'll come to these layers. Okay, later.
Yes. Then we studied double fertilization, in which we studied male nucleus within egg cell forms a zygote. Then zygote forms embryo.
Yes.
Then it is a first scenario, and the second scenario, uh, male nucleus polarizes with
These are two events happening
two
at the
events.
same time, right? That's why they are called double fertilization. There
Yes.
are two fertilizations happening at the same time in a plant.
Hmm.
One gives rise
So
to zygote, which develops into embryo, and
okay.
another gives rise to endosperm. Yes,
And
continue.
okay. To so it is known as triple fusion, basically. Okay.
Yes, because three nucleus are fusing. And what was endosperm?
Endosperm is a inner part or inner layer of the fruit, or we can say
No, not fruit. It's part of the seed,
seed. Yeah, seed.
not a part of the fruit.
It's inside inner layer of the seed. Yeah.
And what does endosperm?
So
Why is it there?
it is there to nourish the seed while it's growing. Yeah,
It's to nourish the developing embryo. Okay.
development to it. Yeah, yeah.
Okay.
And we included the example coconut, water of the coconut. Yeah,
That is the
remember.
liquid, it's a liquid endosperm. So what is special about coconut
Yes,
is it's a liquid endosperm. Is it clear?
yes, sir.
Yes. Okay. We also talked about dicots and monocots.
Yes.
Remember? Yes. What is dicot and monocot?
Dicot means there are two embryos in the same shell, in the same seed, and
There are
I'm
embryos.
yeah, two embryos. Sorry.
Two cotyledons.
Two cotyledons in the same embryo.
In the
Embryo, yeah.
seed.
Seed. Sorry,
If there are two embryos, then there will be two plants, right?
two plants. Yeah.
Does it ever happen that you sowed one seed and there are two plants coming out of it?
No.
So embryo has to be one. Cotyledons has to be two. So it can open from the middle, like I have shown here. Look here. This is a gram seed that we can split from the middle and we can open it, right?
Yes.
We'll now, in today's class, we'll come to the deep structure of these monocots and dicot seeds. But dicot means dicotyledon. There are two cotyledons present, and therefore in the cotyledon, endosperm is also present. In monocots, there is only one cotyledon present. Okay, so it cannot open into two.
Okay.
Okay.
So yeah.
And then in the last class, we also discussed that we will be doing structure of seed, fruit, which is true, false, and parthenocarpic in the next class, which is today. So we'll do it. Okay. So let's move forward where we left. Okay.
Okay.
Okay. So before we move forward, Zed, let's revise everything with respect to fertilizing that has happened. So last thing is, now after fertilization, we will be forming seeds and fruit. That is the third step. There are three stages. First stage is pollination. Second stage is formation of zygote and embryo, development of embryo. Third stage is formation of seed and fruit that we will do today. But before that, look at this. Is this visible to you?
Yes, sir.
Yeah. What color should I use? Let me use blue here. So let's start from this. This is the first stage, okay? Flower. So if you cannot draw the figure, you still have to make this cycle. So just write flower. Do not draw the flower, but write flower. Okay. Let's start from the first stage, which is flower. Now we know that flower has. See, this is the male structure, correct? And this is the female structure, correct?
Yes.
This is a bisexual flower. Now from the flower, there are two arrows going. So one arrow is going to the male structure, which is stamen. The other arrow is going to the female structure, which is pistil or gynoecium. We can also call stamen as androecium. So all these names are now common to you, right? You do not, it's not confusing.
Yes.
If I use gynoecium, pistil or carpel, it's the same thing. If I use stamen or androecium, it's the same thing. So let's begin with stamen first. So stamen contains anther, right? And I told you anther could be bilobed or monolobed. So this is clearly a bilobed anther, which means total 4 are there, correct?
Yes, sir.
And microsporangium is the word used. The word microstat is used for the male. So microsporangium gives rise to the male gametes. Or what is the gamete equivalent in plants? Pollen. So this is microsporocyte. The word cyte means, see, all these words are very, very simple. The word cyte means
Site.
cyto.
Okay. Cyto. Cytoplasm.
Cytokinesis, cytoplasm, cytosol. The first word that I told you when I asked you to make terminology section is the word cyto. The word cyto means cell. Look at your terminology section and write it down.
Yes, sir.
Okay, the word cyto. Cytology. Cytology means?
Study of cell.
Yes. And microsporocyte means those cells which give rise to the microspores. And microspores are pollen. Okay. Megaspores are ovules and microspores are pollen. So inside these stems, inside the theca, you know about theca. Any word that I use, if you are not confident about it, please stop me, okay?
Okay.
And do you see this blue and red color? So wherever you see blue, this indicates the diploid stage. See, it's written here. Red is haploid and blue is diploid. Okay.
Okay.
Anther is diploid, this flower is diploid, and the microsporocyte is also diploid. When the microsporocyte forms the microspore tetrad, this is the first stage where, see, this blue arrow is becoming red. Do you see it?
Yes.
So from microspore to microspore tetrad, from diploid cells, haploid cells are now formed through mitosis. Is it clear?
Okay.
So this is a tetrad. Sorry, meiosis. Through meiosis, the microspore tetrad is formed. Now, as I told you, there is generative and vegetative cell, but only the generative cell takes part in fertilization. So we will study about the generative cell here. See, this is the generative cell, and this is the tube cell, which is the vegetative cell in a pollen grain. Now, from this part onward, when we make pollen grain, we know that pollen grain is strictly haploid, correct?
Yes.
This is the what we say, the borderline, the transformative stage. Now, from this pollen grain, the tube nucleus gives rise to the pollen tube. Can you see this pollen tube growing?
Yes.
Okay. Now, this part where we are actually making pollen and pollen grain is called a microgametophyte. What is it called?
Microgametophyte.
So there are two stages in a plant's life. One is called gametophyte and the other is called sporophyte. Gametophyte is when gametes are being formed. Sporophyte is when diploid body is being formed. Okay.
Yes.
So this is microgametophyte germinating pollen grain. So you see this structure, a germinating pollen grain with a pollen tube and sperm. The word sperm here is used for the male nucleus. Okay. We do not call it sperm in plants, but it has been used here to make you understand. Is it clear?
Yes.
Let's stop it till here. So we reach till microgametophyte is formed. Okay. A haploid structure.
Yes.
Now let's come back to flower and go the other way. The other, if it's a bisexual flower, it also has a gynoecium or a pistil as we know it. Now inside that, this part is called stigma. This part is called
Style.
and they have marked ovary.
Ovary and
Inside the ovary there is a ovule that they have shown. Okay, so ovary and ovule they have shown. Now, just like microsporangium was there for the tissue which gives rise to male gamete, there's a tissue called megasporangium which gives rise to the ovule. Okay, the word mega is for females, so it is an immature ovule. Then when it starts maturing from this, so these are the structures of different parts of megasporangium. It has a stalk, it has a micropyle. You know what is the micropylar end, right? I told you already. So somewhere towards here will be formed your synergid cells, the egg cell, and the filiform apparatus, right? Towards the micropylar. Yeah. And somewhere from here only the pollen tube will enter after it is mature. So currently it is an immature ovule. So it has nucellus, megasporocyte. Just like microsporocyte, there is a megasporocyte. What does megasporocyte means now? You already know.
Mega represent female structure. Sporocyte means spore and side means the cell. So females.
Megaspore means ovule. Okay. Ovule. Microspore means pollen. So megasporocyte is the cell which gives rise to ovule later on. And as we know, inside the ovule, what is present? Embryo sac. So you see this megaspore. Can you see that in that megaspore there are multiple nucleus? They are all starting to align now. And this is the transformative stage where diploid 2N is now converting into haploid. So meiosis is happening here also. After megaspores are formed, they make megagametophyte. Just like microgametophyte, there is a megagametophyte, which is the embryo sac. Okay. Now, this embryo sac does not have a 7-celled and 8-nucleated structure yet. But after some time, see the mature ovule. Does it have a 7-celled and 8-nucleus structure?
Yes.
This is a true mature ovule. So you see antipodals, you see polar nucleus, you see synergid, and you see the egg cell. Okay, correct?
Yes.
Everything. Now let's come to, so this was stage 1, flower. Then flower made all this, either a male or a female gamete, and we reach till a microgametophyte and a mega gametophyte, which is a mature ovule. Now what will happen is both of them will meet together at the event of pollination, correct?
Yes.
The pollen will formed on the will fall on the stigma, and already this mature ovule is present here. Can you see this?
Yeah.
This is the mature ovule. Here, the yellow in color. They have marked it yellow. Now, after pollination, there is fertilization, and after the fertilization, this embryo sac develops into seed. You see that?
Yes.
The seed. So there are two major structures, three actually, of a seed. One is the seed coat for protection. One is the embryo, that is the actual thing that will grow into a new plant. So we have protection, and we have the baby for which we need the protection. Last thing we need is endosperm, right? So inside this seed, the third thing which they have not mentioned will be
Cotyledons and
endosperm.
endosperm.
So a seed. Yes, am I audible?
Yes, sir. Can you excuse me for 1 minute?
Yes.
I have to change that.
Yes, please. When you are back, just let me know. Okay.
So can you hear me?
Yes, is there?
Yes.
Shall we begin?
Yes.
So a seed has three major things. One is the seed coat, one is the embryo inside, which is protected by the seed coat, and third is the nutrition for that embryo, which is endosperm. Is it clear?
Yes, sir.
A, B, and C. These three things has to be present in a seed. Now this seed, when it is in the soil, when we put that seed in the soil, it starts germinating, and after germination, it forms a new plantlet, right? A new seedling. You see this structure? And you
Yeah.
see this is sporophyte, a young sporophyte. So sporophyte is always diploid, 2n, and gametophyte is always
Haploid.
So will you remember that for every plant, gametophyte is haploid and sporophyte is diploid.
Yes.
And then this sporophyte again makes flower, and this cycle continues. Is it clear?
Yes, sir.
So even if you cannot make these structures, like which things which are complex, just write the name, and please make this flowchart right now in your copy. I'm giving you 4-5 minutes. Okay.
Okay, sir.
Note it down and let me know when you have noted it down. Let me quickly grab water for myself. Okay.
Yes.
Yeah. You finish it and let me know. Just do. Yes, Zed, I'm back.
Yes, sir. Still making.
Yeah. Take your time.
Sir, can I make it after the class? Is it possible?
Can you access it after the class?
Yeah, I have just took the picture.
Okay, you took it? Screenshot?
Yes. Hmm.
Perfect. Okay, so let's move forward. So you have to make it in your copy in your own words so that you understand the
Yes.
and it continues. So this was to explain and make you understand what is megasporangium, microsporangium, whether they are haploid, diploid, at which stage do they come, and most importantly to let you know about gametophyte and sporophyte. Okay.
Yes.
Okay, perfect. So now we'll move to write down the third stage in sexual reproduction of flowering plants, write down formation of seeds and fruits.
Okay.
Formation seeds. So let's first start with seeds, and we'll also write about fruits. So first seed. So what is a seed? We know by now that there are two types of seeds, monocot and dicot, right?
Yes.
So first let's make a monocot seed. So we know that a monocot seed only has one cotyledon, correct?
Yes.
Draw it a rough monocot seed for you because it only has one cotyledon. This is, let's say this is the one cotyledon. Now the major part of the seed of course has to be endosperm, correct? So an easy way to
Yes.
make a monocot seed is to first divide it into two, where one part is endosperm. So this part clearly, bigger one, is endosperm. Okay, the nutrition for the seed. Now because it has only one cotyledon, it will not have as much endosperm as a dicot seed can have, correct?
Yes.
But what is important to understand here is, now one interesting thing that monocot seeds have is a layer which actually surrounds this endosperm. So this layer surrounds the endosperm, black one, is called alluron layer. Alluron layer. Okay.
Okay.
And the top thing here, like this outermost course, above this will be the seed coat. Okay.
Okay.
Now the bottom part, which is present, will have the embryo. The second important thing is embryo, correct? Endosperm, seed coat, and the embryo. So this has the embryo, and in an embryo, first this region here is called scutellum.
Uh,
Yes, sir. Ask me.
yes.
Yes. Scutellum, and then there is this embryo present in this region, which grows. Now in this embryo, the topmost region here, which I'm marking here, and then there is this bottommost region. So the first part, so scutellum is above the embryo. Okay, let me mark this thing better. It's this region. Is it clear? It does not touches the embryo.
Yes.
First topmost part that touches the embryo in a monocot is actually called coleoptile. The bottommost region is called coleorhiza. Okay. I think it's double R. Please check the spelling once. I think it is double R. Double R H I Z A.
Yes. Yes.
Beneath coleoptile, And so coleorhiza is followed by, on the bottom side, another layer of cells which forms the radicle, because on this side roots will be formed. Is it clear?
Yeah.
And on the other side, beneath the coleoptile, what will you find? The tissue which will give rise to shoot in the embryo. And what is that called? Plumule. Remember?
Yeah, please.
I told you above. So this is basically the overall structure in a monocot. There is endosperm on the upper half. Like major part of the seed contains endosperm. And endosperm has a alurone layer around it. Then there is, in the bottommost part, there is scutellum, coleoptile, and coleorhiza, which are followed by radicle and plumule. Okay, coleoptile is followed by plumule, and coleorhiza is followed by radicle. Is it clear?
Yes.
Now this part, this whole part here, all these things, is the embryo. Is it clear? The third structure, third important thing: endosperm, embryo, and seed coat. Is it clear?
Yes.
For a monocot. Now
Yeah.
let's make a dicot seed. Now what is the difference between a monocot and a dicot seed? The very clear cut difference.
So monocot is having one cotyledon and dicot is having two cotyledons.
So first, when we'll make the structure, we'll make, yes. So it has, so this is an example of, we can say, a corn. A corn seed looks like this, right?
Yes.
If it is a rice seed, the structures will be same, but the shape will be a little elongated, like a rice grain.
Yes.
Now here, let's make a bean, a kidney bean seed, which is a dicot. Let me write first. Did you, have you finished drawing this?
Yes. Yes. Yes.
Along with me? Yes. Now we're going into the details of the dicot seed. As we know, there will be two cotyledons now, right?
Yes.
And these are the two cotyledons in a kidney bean. Both have to be equal. Just pardon my drawing. Now in this, where do you think the first thing, of course, is the seed coat? The outermost part is the seed coat. Then we are left with endosperm. So where will be the endosperm present?
Topmost.
In both the cotyledons, there will be endosperm. Okay. So this as well as this, both are
Yes.
endosperm tissue, and we call it cotyledon, right?
Yes.
Cotyledons. Now the third thing is embryo. Now where is embryo and how is that embryo arranged in a dicot seed? Is like, it's not like in all the dicots there are two embryos. Remember you said that there are two embryos in dicot seeds, correct? You said that.
Yeah.
No? But the point is, then two embryos will give rise to two new organisms as well. But it is one, but it is like, it is embedded like this. So if you see, it's somewhere around here. It's elongated. It's present here, and again it has some structures. Okay. So what I'm making here is the rudimentary leaves. It is inside. This is how it grows. Have you ever seen it growing?
Yes.
Now let's mark these structures. The structures are, the topmost part is called epicotyle. What is called?
Epicotyl.
What does the word epi means?
Ending post.
Epi.
Top. No. Epi.
Our skin is called epidermis. So what
Epidermis. Yeah.
what,
Top. top, upper part.
Above. On.
Above. On.
So it is epicotyle. It is on the cotyledon, right?
Yes.
Then followed by it is called hypocotyle. Is it clear? Epicotyle,
Yes.
hypocotyle. Now we know that this epicotyle and hypocotyle, beneath this structure gives rise to radicle. So radicle gives rise to shoot, right? There is no such division of epicotyle and hypocotyle in a monocot because there is only one cotyledon, and that is all it is. So it cannot open, and we cannot distinguish it between cotyledons and the embryo. So that's why there is a top and bottom arrangement. Here the embryo is present squished, sandwiched between the two cotyledons. Is it clear?
Yes.
Yes. So this structure, the hypocotyle, gives rise to the plumule, and beneath that the epicotyle, the radicle is there, which gives rise to the shoot. Sorry, the root. Is it clear?
Yes.
No doubt.
Not.
Okay. Very good. So monocot and dicot seed is clear to you.
Yes.
Also drew both the structures.
I have to do this dicot.
Just do it for dicot, and this is the embryo. So you see, in a way, if you mark where is the embryo is present to the exact length of the seed, right?
Yes.
Because it's squished and sandwiched. So this is the embryo. I have color coded the orange one. You see that the length of the seed is actually the length of the embryo also. But here the embryo is smaller than the seed. Is it clear?
Yes.
Okay, draw this and then let me know, and I'll move forward.
Sorry. So I want to know, like, hypocotyle. What is this meaning? Like hypo.
I'll make you write all this thing. I'll dictate everything for boards. Don't worry. Just finish this structure and let me know. Then we'll come to the structures.
Yes, sir.
Is it done?
Yeah.
So write down. Start writing. Parts of a seed. Parts of a seed. Question can be asked in the entrance on any of the part of the seed. Also in boards, they can ask, draw a monocot seed or a dicot seed and explain its part or mark its structures, right? So these kind of questions come from this part. Is it okay, Zed?
Yes.
Would you like to write the questions?
Yeah.
Yes. Just write down. Draw monocot or a dicot seed. Mark its structures. And explain the parts of a seed. And explain the parts of a seed.
Okay. Sir.
Write down parts of a seed. So seed is the most important part of a sexually reproducing plant. Seed is the most important part of a sexually reproducing plant, as they contain embryo inside. As they contain embryo inside and are capable of giving rise to new plant. Just write down one more question which comes in boards. What is germination? You know what is germination?
Seed germination. Yeah.
Yeah. So they ask
formation of we can say, like, formation of new sporophyte, like
new plant.
from, yeah.
Normally defining it, even if you don't use the word sporophyte, it's fine. So write down what is germination. Write down the answer. The growth of a new plant. The growth of a new plant from a seed. From a seed. Growth of a new plant from the seed under favorable growth conditions. Under favorable growth conditions is known as germination. Is known as germination. Is it clear?
Yes.
Now, as you can also see in these diagrams, I told you, write down. A seed has majorly three parts. A seed has majorly three parts. Now, these three parts always have to be there. First is the seed coat, correct?
Yes.
Most important, otherwise there will be no protection. Then the endosperm. If this is not present, then there will be no nutrition for the growing embryo. The third part is the embryo itself. These are the three major parts of a seed, whether it is monocot or dicot. So you can write for both monocots and dicots. So above in the diagram, I showed you the difference, correct? Here we'll talk about the important common things.
Okay.
Now, in class 11, let's go back to class 11th a bit. Did you remember studying imbibition, osmosis? In the chapter osmosis,
Yes.
there is a term called imbibition. Can you tell me what is imbibition?
So if I'm not wrong, it's like
I'm writing
it's
it. This is the term imbibition. What is it? Just allow me one moment. Okay, think about it. I'll be just writing. 1 second. Yes, Zed, I'm back. You there? Remember what is imbibition?
no.
What is imbibition?
I thought like I no.
You remember studying that solids like wood can absorb water, and when they absorb water, their volume increases.
Yes, sir.
In earlier times, people could use wood to break boulders. You can even break a mountain by just wood thing. So if you see a very big rock which you cannot break, and it will take lots of hard work and tools and lots of people to break, what you can do with the time, if that rock has cracks, any cracks, just put wood chips in that crack and leave that rock for the monsoon season. So when monsoon will be there, or pour water on that wood, so that wood will imbibe water and will expand in volume. And when it will expand, the pressure will be so much, it will make the crack split more. And over time, slowly, you can actually break that boulder. That's called imbibition. That's called the power of hydrostatic pressure. Remember?
Hydrostatic pressure. Yeah.
Yes. Hydrostatic pressure. So when there is water somewhere, it creates a pressure. When we get some swelling in our body parts, let's say our fingers or toes or in the tonsils or anywhere, that swollen part feels very pressurized, right? Hard.
Yes.
Because water accumulates during swelling, and a hydrostatic pressure also causes pain because it's a pressure from inside. That's why things that get swollen are also painful. Remember? So this was
Yes.
now the seed coat is very important structure, not because it protects, not only because it protects. And also, let me ask you one question. Seed coat is hard, right? It's a hard structure.
Seed coat. Not every time. Like, yeah.
Yeah, it is a hard.
Uh,
If it is not hard structure, how will it protect the seed? So it is a
yes.
hard
It's
structure.
not.
But the
Hmm.
problem is, if it is a hard structure, then how does the embryo come out of the seed? In
Same procedure. Imbibition.
no, yes, right. So when seed coat imbibes water, that's why to grow, to germinate the seed, it is very important to put that seed in soil, because soil will have moisture and water, and the seed coat will imbibe water, and when it will imbibe water, it will break open, correct, because of pressure,
Yes.
hydrostatic pressure. So when it will break, the embryo can now come out. Till the time the seed does not find water, so finding water for a seed is very important, not because the embryo needs the water, not only because the embryo needs the water. The developing embryo starts needing water later on. First it has to come out of the seed, correct? So first requirement of water is to break the seed coat. Is it clear? This is an entrance question, write down.
Yes.
First encounter of a seed with moisture is not for embryo, it is for the seed coat to break. Right on. The first encounter of a seed with moisture, the first encounter of a seed with moisture bring out the seed from dormancy. You know what is dormancy? A dormant,
A stage of rest.
a stage in which it cannot germinate. So a seed which does not find moisture will never germinate. You can understand now why. Because the seed coat will never break and there will never be a chance for the embryo to come out. Is it clear?
Yes. So I have a question. Like inside the seed coat, uh, is there like air can go inside, like, or it's
Air.
a sealed? Yeah, air.
Yeah, air, even water can go inside. No, that's why when what they found moisture, the water does seep inside slowly. If it's not impermeable, things can go inside. Air and water, moisture has to go inside, seep inside, so that it can start swelling the seed coat and it breaks.
Oh,
Okay, it's not a airtight cavity, it's not like that. But till it does not find moistures, it is a dormant seed, it cannot germinate. We
oh,
can grow seeds without soil, but we cannot grow seeds without moisture. Do you understand? There is something called hydroponics. Have you heard of this word? Again, class 11th or 10th, I'm not sure, some junior class. Hydroponics, aeroponics. Hydroponics is a technique where without the soil you can grow and germinate a plant.
in the like example is like rose.
No, so there is a pipeline in
Mhm.
which you put
Yeah, yeah.
plant.
In school we made it. Yeah, in school we made it.
Yes, and there are inside. There are multiple plants like this. You understood what I'm trying to say?
Yes, yes.
And from this pipe, water is flowing, and that water contains nutrients and growth supplements, whatever is required. But there is no soil in this whole setup, but still the plant grows. Make sense?
Yes.
That is called hydroponics. Okay,
Okay.
but we need water or moisture for that. So write, you written down. So for, let's come to the first point. Write down seed coat. Let's write something important about seed coat. First point, make bullet points and keep writing. Okay, short things. I will, no need to write full sentences and a paragraph. First bullet point, protects the internal parts of the seed. So seed coat protects the internal parts of a seed. And you ask a very good question. See, if the seed could not engage or communicate with the environment, if it cannot know when air is around or moisture is around or water is around, then it will never come out of dormancy, no? So it has to be able to sense moisture in the air or in the soil, correct?
Yes.
Yes. So that's why these things can go inside, air and water can go inside. So it protects. Yes, Zed. You have a question?
No, finished.
It protects the internal part of the seeds. And seed coat itself has two layers. Write down, it has two layers. The outer layer is called testa. Seed coat is double layered. The outer layer is called testa. It is a thick layer. And the inner layer, this is the word that you might have read in book called tegmen.
So actually, you only told like both first, like when we were like doing something, you told these words, testa and tag.
Yes, and I told you that
Yeah.
at the right moment I will tell you what are these. So when we are going through a story, there is a right moment for things to learn, right? Now you are understanding it better. So seed coat,
Yes.
two layers. Outer is called testa, which is a thick layer. Inner is called tegmen, which is a thin layer. So write down seed coat. Seed coat, next point, prevents the seed from dehydration. So there is some amount of moisture present inside the seed also, correct? The embryo, the endosperm, they have some moisture because they are cells, they are living cells. They will be having cytoplasm
Yes.
and moisture. So if a seed is just lying outside in the sun, that seed should not get so much dried up and dehydrated and desiccated that it dies. So the seed coat prevents loss of water. Loss of water, in brackets you can write dehydration of the seed. Seed coat prevents loss of water, dehydration of the seed.
Okay.
It also prevents entry of parasites. So it also does not allow anything to come in, because endosperm is a nutritious tissue, right? So something can come in and start eating the seed. Sometimes when it failed to do so, you will see that some seeds look perfect from outside, but when you open it, they are gone from inside.
Yeah.
Yeah. So this is where there would have been some crack in the seed coat and the parasite or the insect entered. So it also prevents entry of parasites.
Okay.
Next point, seed coat also prevents germination. So there is a signaling happening in the seed. Embryo always wants to come out when the environment is favorable. Otherwise, it's a waste, right? So if let's
Yes.
say the embryo was protected inside the seed and outside environment is hostile, it is 50 degrees heat, burning heat, an embryo just came out. The moment it will come out in this heated environment, it will get dehydrated and it will die, right?
Yes.
So the seed is a waste. So seed coat also prevents germination in unfavorable environmental conditions. It also prevents germination in unfavorable environmental conditions. Okay.
Okay.
Now there's one question that have been asked about the seed in entrance. In seed, there is a structure called in the seed coat. There's a structure called hilum. Just write this point and remember it for your entrance. The word, there is a hilum or hilum.
Yeah, in this that
The seed,
embryo sac it is there. Yeah. Embryo sac, phylum.
The region that develops when embryo sac ovule develops into a seed. This is the region from where the embryo starts coming out from. You understand? It's a small
Yes.
pore-like structure. And hilum is equivalent, write down hilum. Hilum is a visible structure in seed coat. Hilum is a visible structure in seed coat and is equivalent to, equivalent means when we are comparing certain structures, we say that this structure in this organism is equivalent to this structure in other organism, correct?
Yes.
Like we say that the flippers of the whale, the first flippers of the whale is equivalent to a human hand, because whale is also a mammal and human is also a mammal, correct?
Yes.
So write down the hilum is equivalent to the navel. Navel. In our bellies, there is a hole, navel, you know, in humans.
Yeah, this.
That is the structure where umbilical cord was attached when we were inside the mother's womb. Okay.
Yes.
About this, about the umbilical cord, we will study in the next chapter that I'll start from the next class onwards, which is human reproduction. Okay. So where there in towards the very end, we will read about umbilical cord and why is it important as a tissue, what it's a magnificent biology. But hilum in a seed is equivalent to navel in humans because that is also a hole and our navel also looks like a hole throughout the life. It's not a proper hole because there are tissues that now have filled it, but you can see it as a hole, right?
Yep.
Write down it is equivalent to navels in human where umbilical cord is attached.
So it is NAVEL or NAVAL?
It is NAVAL.
Okay.
Navel.
Okay.
Or it is NAVEL. Now I'm not. Just check it, okay? Somehow,
Okay.
because they will ask. If they'll ask you, they'll give you options. But you understood which thing I'm talking about, right? The hole in the belly button, we call it.
Yes. Yes.
So where umbilical cord is attached. So this I just saw in one question. Now we cannot go and start reading about hilum completely, but we should know this, okay? So seed coat is clear?
Yes.
Perfect. Now let's come to the second part, which is endosperm. Write down endosperm. I also have told you about endosperm above, right? When we discussed coconut thing. So just write down endosperm has nutrients stored in it. Just bullets, right? Keep writing bullets. Endosperm has nutrients stored in it. I also told you that majorly it is starch and proteins, right?
Yes.
Carbohydrates and proteins to support the embryo, the developing embryo. See, when anything is developing, starch and proteins become very important because lots of structures are being made, correct? Yes or no?
Yes.
And starch is required as an energy, which we burn, like glucose, which we can burn or oxidize during the process of respiration to get lots and lots of energy to do anything. And protein is used as the building blocks. Amino acid build the cells, right? Mostly everything.
Yes.
So we need raw material for building block and we need energy to do that. So when we are making a building, what do we need? The raw material and labor, people who can do it, energy, right? Then when
Yes.
it's complete, we need other things to just fine tune the structure. So, but majorly what we need is in bulk is we need everything. We also need lipids because you know all our cell membranes are made up of lipids. It is lipid bilayer. Lipids are a
Yes.
kind of fats. So we need fats, we need vitamins, minerals, everything. But in bulk, we need starch and protein that endosperm contains. So write down next. It is located below the seed coat. Endosperm is located below the seed coat. Write down in monocots. In monocots, endosperm is the major portion of the seed. In monocots, endosperm constitutes the major portion of the seed, also which is visible here. Can you see this?
Yes.
That is endosperm. So in monocots, endosperm constitutes the major portion of the seed. Whereas in dicots, this is another difference between monocots and dicots. Whereas in dicots, endosperm is utilized in the embryo development. Endosperm is utilized in the embryo development and is absent in a mature seed. And is absent in a mature seed. Why I asked you to write this? Because a question, if it comes, see, rice and pulses, both are seeds, correct?
Yes.
Rice. But what do we say? Rice is a good source of
Protein, carbohydrate. Sorry, starch. Starch and yeah.
Yes, starch. We do not get much protein from rice. We get starch from rice. But pulses are another seeds, for example, beans, right? But they are a good source of
Proteins.
proteins. Then both are seeds. Then how does this difference come? Because rice is a monocot, right?
Yeah.
And they have intact endosperm in the mature seed, correct?
Yeah.
Yes. Pulses are dicots, and in a mature seed, endosperm is absent, right?
Yes.
Because it gets used up in the embryo development, even when the embryo is inside the seed only. So this is where the difference comes from. So do you understand now why rice is a good source of starch being a monocot seed, but pulses being dicot seeds are not a good source of starch but are good source of proteins, because all the starch is used up. Okay.
Yes.
Okay, very good. I think, and then again, coconut is a liquid endosperm. That is a very famous thing. Done.
Yes.
Now let's come to embryo, third part. Write down embryo. The main thing. This we will study in detail. Embryo. Now, embryo itself is a very complicated structure, because here I have written different parts of the embryo itself, right? Epicotyl, hypocotyl,
Yes.
coleoptile, all these things. So write down. Just like seed is the most important part of a sexually reproducing plant, embryo is the most important part of a seed. So this question has come in a fill in the blanks type pattern, where they say that dash is to the sexually reproducing plant as dash is to the seed. So what is the first dash? Dash is to the sexually reproducing plant as dash is to the seed. So in the first dash, you will write seed. Seed is to a sexually reproducing plant as embryo is to a seed. Do you understand?
Yes.
Write down embryo. It is the most important part of a seed. It is the most important part of a seed. Yeah. Written?
Yes.
Write down it's diploid. Oh, sorry, I forgot. So endosperm is triploid. Just mention it there. Endosperm is triploid. Above, write down endosperm tissue is a triploid tissue because it is a result of triple fusion. Make sense? Everything is clear?
Yes.
Write down, and embryo is a diploid tissue, and it has four major parts. Four major parts. An embryo, which is a diploid. So write down the first part, as we know, is called epicotyl. Okay. You were asking me what is hypocotyl, what is epicotyl, right?
Yes.
First is epicotyl. Second is hypocotyl. Third is radical. And then fourth is your cotyledons. Okay. These are the four parts.
Okay.
Write down in an embryo. Epicotyl. Epicotyl is the small shoot. Okay. So did I mention it wrong above? Yes, please. There's one correction there. Do you see the structure?
Yes.
So I have mistakenly mentioned plumule as hypocotyl. It is epicotyl. Okay. This is plumule. Okay. Correct it in a dicot seed.
Yes.
Let's come back. So write down epicotyl is a small shoot that ultimately gives rise to the whole shoot system of the plant. So in short, epicotyl is the plumule and gives rise to the whole shoot system. Is it correct? Is it clear?
Yes.
So when it grows, when it develops, it first develops as just one or two small leaves and a green little stem which comes out of the seed. You must have seen germination, right? I don't need to tell you.
Yes.
Then next come to hypocotyl. Now this is a very interesting and important structure, hypocotyl. You know, there are two types of roots also when we studied roots. One is called primary root and the other is called secondary root. Any idea?
Yes.
Yes, sir?
Primary roots are like which are developing new roots, which are we can say on the
From which
edge of the like root system, and the secondary roots are like which are developing from that roots, like small roots. Yeah.
root nodules.
Nodules.
Yes. Now a hypocotyl is equivalent to a primary root, but it's not a true embryonic root. But what happens is, why it is important is because when you put a seed in the soil, do you worry about the seed's orientation?
Mm, orientation, like it's symmetry, like it's downwards or upwards, like that.
Do we worry about such things?
No.
But always when the plant germinates, the shoot comes towards the top and the root goes towards down, right?
Yes.
We say that there is the reason is because shoot is attracted towards the sunlight, but shoot is attracted towards the sunlight after the shoot is formed, right?
Yeah.
So you need to have chlorophyll and you need to have all that signaling in the shoot so that it can figure out where sunlight is. But it is also possible that a plant starts growing diagonal, horizontal, or does not, you know, let's say, look at this seed. So this is a gram seed I am making again. See, this is a seed, okay? You see, in a gram seed, there is something at the top, like a blob here, notch, right?
Yes.
Not a perfect gram seed I made, but you can understand, correct?
Yeah.
Now let's say from here, the part which grows goes up and forms the leaves, and then roots goes down, let's say. But inside the soil, this seed could have also have been present like this, no?
Yes.
So will the shoots, when it will start emerging, it will go all the round way and then go up into the soil, or like up outside the soil?
Yeah.
No. And then the roots will grow and it will go all the way down again. It is too complicated, no?
Yes.
Instead, what the seed does is it reorients itself before germination starts. And this reorientation happens because of hypocotyl. Is it clear? So it's
Yeah.
a like tissue which first grows. It's hypocotyl is the first thing that emerges out of the seed even before epicotyl or radicle comes out and aligns the seed properly so that plumule goes upwards and radicle goes downwards. Is it clear?
Yes.
So write down about hypocotyl. So do you understand that there is a reorientation happening of the seed? It's an intelligent system, no?
Yes.
Biology is very, very brilliantly intelligent. Not, and whenever I say intelligent, do not think of intelligence from a human perspective. Intelligent as a system, correct?
Yes.
Which we also known as intelligently designed system or evolved system. Okay.
Okay.
Okay. Write down hypocotyl. Hypocotyl is the primary root-like system. Hypocotyl is the primary root-like system that emerges the first during germination. Hypocotyl is a primary root-like system that emerges the first during germination. It helps in orienting and anchoring the seed. It helps in orienting and anchoring the seed firmly or strongly in the soil. It helps in orienting and anchoring the seed firmly and strongly in the soil. Is it clear?
Yes.
This is the first to emerge. Primary root. Okay. Now let's come to radicle. Write down radicle.
Okay.
Yes, Ed. Question?
No.
Radicle is simply, you know that radicle gives rise to root system. Write down. It is a small embryonic root. It's a small embryonic root which later on gives rise to the plant root system. Which later on gives rise to the plant root system. Which later on gives rise to the plant root system.
Okay.
Okay, Zed?
Yes.
Now, cotyledons, you already know. Last thing, cotyledons.
Yes, sir.
Write down, they emerge as, when an embryo is growing, these cotyledons, write down, they are tiny first, before emerging, write down. Or you can write down, they emerge as tiny, sorry, you know, when I dictate, I think what is the best way to make it right. So they emerge as tiny fleshy like structures from the soil. So have you seen a plant germinating? You will always see that from the soil. So this is the root system. Then there's a green small stem, and then there are two leaves like structures, which are not leaf actually. Leaves are formed later on. But these two structures are there, right? They are cotyledons.
Yes.
So they emerge as tiny or fleshy leaf-like structure from the soil along with the seedling when it is growing. Along with the seedling when it is growing. And you can write it contains nutrients. Contains nutrients to support the growth. It contains nutrients to support the growth. I've already told you either they can be monocots or there can be dicots plants. Okay.
Okay.
So is the seed thing clear to you?
Yes.
No confusion, no doubt in the seed.
No.
Okay, brilliant. So next, write down fruits, the last topic for today. Fruits. Even if you are not able to finish it, we'll just try to. So in the next class, which is next Monday, right? I want to start with human reproduction for your batch. Okay.
Okay. Sir.
There'll be some homework that I'll be giving you today that you have to. So for in the next class, probably we'll be having like normal length 2-hour classes. So we'll spend half an hour to wrap this sexual reproduction in plants first, and then spend the rest 1.5 hours on human reproduction. Okay, majorly, because human reproduction has a lot that we need to cover.
Okay.
And keep revising. You know, as the syllabus will move forward, it will also gain pace. And I hope you are not finding it very overwhelming. Okay.
Yes.
So fruits. What are fruits? Any ideas, Ed? How will you define a fruit?
Fruit.
Fruit is basically defined as a ripened ovary. Remember, I told you that
Yeah, ovary develops into fruit. Yeah.
And ovaries are the future fruits.
Yes.
Write down. Fruits are defined as ripened ovaries. Ripened ovaries. Now, if I go back to the structure on the top, where is the structure? Yes. Look here. Come back here. So see this ovary, swollen structure, Zed? And I told if there is some tissue around which is called thalamus, then there are some tissues which are called peduncle and everything, receptacle, things like that, right?
Yes.
So this ovary, when it ripens, so inside the ovary are ovules, so it becomes seeds, and the ovary becomes future fruit. Okay.
Okay.
Now there are two things that you need to know in fruits. One is, so true fruits are the ripened ovary. Can you give me an example of true fruits? The definition that I just gave you was a definition for true fruits, because they are the fruits. If something is true, we do not call it true, right? We just call it fruits.
Yes.
True fruit. Example. Any
example of fruit? Like I have an example of false fruits, but I'm not getting true fruits.
Okay. Write down mango. Very aptly called the king of fruits because it is a true fruit. You know, king can only come from the true things, right? You cannot make a false thing.
Yes.
King of something. So mango, grape, even your maize. You know, maize or corn. You know, maize.
Yes.
They are all true fruits. And so they are all ripened ovary. They are all called true fruits because they are all ripened ovaries. Now the important thing is, just like you did in entrances also, they do not focus more on true fruits, they focus more on false fruits. Now, what are false fruits? So let me give you, so let's do one thing, make a column in between, so that we'll do in the next class. Okay, let's just understand it here. False fruits are those fruits, write down, fruits which are formed, fruits which are formed from other parts of the plant, other ripened parts of the plant with or without ovary, with or without ripened ovary. Okay, do you understand what I?
Yes.
Sometimes ovary also gets ripened, but other things also gets ripened, but that is also a false fruit, because when what we are eating is not only the ripened ovary. So a true fruit is only that fruit in which only ovary is ripened, everything else is just not there. Is it clear?
Yes.
Yes. Write down in false fruits, write down one more point. It can develop without fertilization. It can develop without fertilization. True fruits cannot develop without fertilization. It can develop without fertilization. And write down examples. So we will write, I will give you examples with respect to the parts from which they are formed. Okay. Right.
Okay.
Write down peduncle. So you know there is a structure called peduncle, the receptacle.
Yeah, inside, like where seeds are present, like there, like, yeah.
Receptacle.
Uh, like in apple, we saw like in the center there is a part where seeds are formed, and there that part is comes.
So this structure is called peduncle. Just above receptacle and below the ovary, there is a structure called peduncle. Just like thalamus, there is a peduncle and there is a receptacle, right? Sometimes receptacle is called peduncle in some books, but you understand it is an extra ovarian tissue. Is it clear?
Yes.
From peduncle rises your, write down, cashew nut. Cashew nut is from peduncle. This is the, you can find one or two more examples. I am remembering cashew nut. Then comes thalamus. Again, you know what is thalamus?
Yes.
The structure that I just showed you above. From
Yeah, apple.
Develops a lot of false fruits. Most famous is apple, pear,
Strawberry.
pear,
Yeah. Apple, pear, strawberry.
strawberry, gourd, gourds,
Gourds. Gaurds.
Gourds. Gau, guard is that the guard that guards your houses or society.
Okay.
Security guard. This is guards, like bitter gourd, karela,
Bitter gourds. Okay.
and then lauki. You know lauki?
Yeah.
Yeah, those are guard.
Got it.
Got it. And then also cucumber. Cucumber is also a false fruit. First, we do not consider cucumber fruit. We call it vegetable, but it's a fruit. It's a false fruit. Then write down pineapple. Pineapple is a false fruit. Pineapple and jackfruit. They both are also false fruit. Pineapple and jackfruit. Now, pineapple and jackfruit develops from inflorescence. The whole inflorescence. Not peduncle, not thalamus, everything. The whole inflorescence gets ripened. or jackfruits. All these are false fruits. Is it clear?
Yes.
Okay, so let's wrap up this. I'll be telling you some just final things that are important for boards. We will be discussing some questions in the next class before we wrap this up. And be ready to begin with human reproduction, okay, in the next class. And I hope
Yes.
others will join also. Okay, any doubts there, or shall we just wrap up?
So like in PT1 exams, like what they can ask like from this chapter.
In
Yeah, exams like in school exams like
school exams, are
yeah.
they half yearly or some major exams, or the unit tests like?
Unit tests like that.
Yes. So for sexual reproduction in flowering plants, you're asking.
Yeah.
Yes. So all these things that we have covered in today's class, plus like majorly they will ask you about the structure of the flower and everything associated with it, the function of these structures. Okay. And then you should focus more on double fertilization. Okay. Pollination and double fertilization. This part.
Okay.
And the egg sac, this embryo sac in detail. Okay. Seed and fruits will just be like whatever we did today, that part will be enough for your exam. No need to go like more than that for school exams, unit tests. Okay.
Okay.
And read NCERT. Anything else there? See you then. See you.
Yes. Thank you so.
Bye bye. Take care.